Okay, this is Dr. Elizabeth Grenier, and this is for Bio 208, Chapter 25, which is your Metabolism and Energetics chapter that goes along with your Chapter 24 material for our Test 3 unit that we are going to be taking. And you can follow along with the slides provided from Blackboard. These may be a little different from the ones that are currently provided. Again, I may have updated them, just a heads up on that. Okay, so let's talk about metabolism. All right, metabolism is what we are calling in our human body all of our chemical reactions. So when you hear in the media that you have a high metabolism, what that really should mean is you have a high amount of chemical reactions that are taking place that are building molecules as breaking molecules. Some of what it's skewed or messed up is that metabolism begins in the media, in the social culture of our society, to mean fat burning, losing weight. And it's not always about fat burning and losing weight. Metabolism also could be for building muscle, growing. And so we normally say kids have a higher metabolism. They need to eat, they need a lot of energy because they're building and growing, as well as breaking down and utilizing different molecules to create their new building and growing pieces and parts. All right. Energetics is the flow of energy, again, as it's changing from one form to another. So remember that energy is captured in bonds. So in glucose, we have energy captured between our carbon bonds. There are 6 carbons, so there's energy captured between the carbon-carbon bonds. So for energetics, what we're doing is we're taking, for glucose, we're going to take some of the energy between the 3rd and 4th carbon, and we're going to harness that energy and make it into some NADH molecules that are going to then take that energy into the mitochondria. We're going to take that energy between those 2 carbons, and we're going to harness some of it to make 2 phosphate-phosphate bonds and create 2 ATP molecules that we're going to net for energy ability. And then later on, what we're going to do with those 2 pyruvate molecules, which are 3 carbons bound to each other, we're going to break one of those carbon-carbon bonds and create more ATP and create acetyl-CoA. The rest of the carbon-carbon bonds, we continue to create more FADH2 and NADH that go into the mitochondria and ultimately lead to those carbon-carbon bonds end up being carbon dioxide, and the energy that was between them ends up becoming our phosphate-phosphate bonds with ATP. So that's more about energetics, about where the energy is moving, where the energy is being basically recaptured or reconstituted into different bonds that may be more useful, maybe more for what the cell needs in that moment or in that time frame for the energy to be held in these bonds and not in these other bonds. Okay, so energetics is a big deal. That's many people who become muscle physiologists, so they do kinesiology degrees, they do exercise science degrees. Sometimes they go into their master's and their PhD more so in the master's level, you start to call muscle physiology, kinesiology, and exercise physiology energetics. So PhDs in energetics might have a background that's very strong in muscle physiology. Okay, so this is kind of from our book. We're going to talk about, again, metabolism. So in our cells, we are going to have reactions that lead to the breakdown of molecules, so catabolism, and we're going to have reactions and chemical interactions that lead to building molecules. So from our digestive system, if we intake proteins, we are going to, through pepsin in the stomach, through chymotrypsin, trypsin, and other pepsidase and protein breaking enzymes, we are going to break those proteins into the individual amino acids. So this is where we get tyrosine. So we can use tyrosine to make dopamine, norepinephrine, and epinephrine. We can also use tyrosine to make thyroid hormones. All right, but other things we can use tyrosine and tryptophan and alanine is we can take those amino acids, bring them into cells, and then as we need to at the ribosome, create message RNA into a protein, we can reconstitute those amino acids with the help of our tRNA into a new amino acid peptide primary structure chain, and then those amino acids are used to fill in where they're required, right? When we eat fats like butter and oils, many of those fats are in more complex forms. So we have to then take them into our body with lingual lipase in our buccal or oral cavity and our intestinal lipase, pancreatic lipase. We create our triglycerides into the glycerol background, the glycerol molecule, and the 3 fatty acids, right? And so those are then bound up with the help of lipoproteins to be more easily carried in our blood supply. And then those lipids can help make econysoids, help make prostaglandin if we need those signaling molecules. They can also come into the cell and again be reconstituted into maybe triglycerides for storage, or they can go through our oxidative processes to create energy. Again, when we eat sucrose, when we eat sugars, many times we're eating sugars that are in the disaccharide form. So the maltose, the sucrose, the lactose. We absorb food in our digestive tract as simple sugars, monosaccharides. So we need to take lactose, sucrose, and maltose and break them down into the simple sugars of fructose and glucose and galactose. So those simple sugars then, especially glucose and fructose, come in, and depending again on the fed state, the fasted state, the amount of insulin, the amount of glucagon, epinephrine, cortisol, many of our hormones, that glucose could be pushed into a stored form, so we create glycogen. It could be utilized to make energy, and then depending again on the nature of things, some of the 3 molecule precursors could be pulled out if we need to make certain amino acids, need to make more glycerol. And again, after we get to pyruvate broken down into acetyl-CoA, if we start to have a really big backup of acetyl-CoA, we can even take acetyl-CoA and create more fats for storage if we need to. So metabolism, again, is based on we take what we need through digestive absorption. We get amino acids if we need to take into the cell and create more proteins. If we need to take some of our amino acids and convert them into other molecules, again, we take in those amino acids and convert them to either become nutrient energy providing molecules or reconstitute them into other molecules that we need. All right. If we need lipids to create steroid hormones, econysoids, prostaglandins, signaling molecules, or we need lipids to become phospholipids, so then we can create more membranes, more organelles. We pull them in. We can build them into those, again, single individual fatty acids into a phospholipid, into a triglyceride. We can also use it to create enzymes to create energy for ATP making. And simple sugar, same thing. Depending again on some of the influences of the state we're in, the hormones available, we can take some of those simple sugars and immediately start to break them down, create energy. If we start to see a backlog at the acetyl, the 2-carbon side, right outside the mitochondria, we can start to convert some of that in certain cells to fat. We can also create some of our glucose into stored glycogen molecules. So metabolism is taking, again, what we have available. If we need to reconstitute it into what we still need, if we have everything we need, either utilizing it and storing it is our next option. All right. So again, for definitions, metabolism is all our chemical reactions, reactions that lead to typical breakdown of organic substances, so liberating energy, releasing energy that could then be captured into phosphate-phosphate bonds are going to be catabolic bonds or catabolic reactions. Anabolism is typically where we start to build molecules. So when we build messenger RNA, build and replicate DNA, when we build phospholipids for making the cell increase in size, making more organelles, we are going to build proteins through, again, transcription and translation. So we say that testosterone is a big anabolic anabolic steroid. It helps us build in muscles, build bigger muscles. Again, what's going on? We're going to, with the help of testosterone, its receptor, go into the cell and build more proteins from more transcription translation. Okay, so anabolism, usually think of androids, anabolic steroids, and our baseball players. Many of them show up at 18, 19, skinny little boys. They take anabolic steroids. They become huge monster men who can crush balls and hit home runs, right? And so that's what Mark McGwire, Sammy Sosa, Barry Bonds, a lot of them have that taint of they took anabolic steroids to help them get bigger, which is part of the reason why then they were so successful at the home runs that they hit in their career. Okay. All right, so metabolism of what are some of the organic compounds that have a big interest in our bodies that we want to focus on. Of course, the monosaccharide glucose is a big one because we are going to take glucose and either make it, so we can do anabolic reactions and make glucose from pyruvate, from lactic acid, from some of our 3-carbon amino acids. We can also take glucose and catabolic, break it down to create ATP. And when we do that, again, we break it down through glycolysis. We break it down to pyruvate, then we further break pyruvate down to those carbon dioxide molecules, liberating the energy into NADH and FADH2. If we want to store glucose, again, anabolic reactions are to put glucose in a chain of glucose, 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 glucose. That can help us create stored glucose in our muscles and our liver as a glycogen molecule. All right, on the fat side, triglycerides are going to be glycerol, which is a 3-carbon molecule we get from the breakdown of glucose or trigger through some amino acid manipulations. And we add on that glycerol carbon a bond to a fatty acid, a bond to a fatty acid, a bond to a fatty acid, or a bond to a phosphate. And if it's a phosphate, it's a phospholipid. We use a lot of those phospholipids to be our membranes, our organelle membranes. And as a cell wants to divide or grow more mitochondria or make vesicles, they need phospholipids. So that helps us there. We also take some of our fats if we're going to store them and put 3 fatty acids on a glycerol backbone, and that's our triglyceride. And that's our stored form of fat, primarily stored in our adipocytes. Adipocytes can be in any connective tissue. In females, we tend to see large adipocyte populations in your subcutaneous or hypodermic areas, and that's going to be then your subcutaneous stored fat. In men, we see a lot more adipocytes in their peritoneum, the mesenteries, the omentums, in that connective tissue in and around your visceral organs. And those adipocytes, when they hoard fat, are known as your visceral fat store pads. Okay. Again, for metabolism, we are constantly making and breaking proteins. Skeletal muscle, if you want to make them bigger, stronger, they need more actin, more myosin, troponin, tropomyosin, nebulin, titin. Well, those are all proteins that we have to create. If you want to make more molecules on your membrane, voltage-gated channels, ion-gated channels, ligand-gated channels, receptors for hormones, receptors for other signaling molecules. Many times those are proteins. Proteins are also many of our enzymes. So if you want to have more enzyme that, like a carbonic anhydrase, well, then you need to create the protein or carbonic anhydrase. So then we can create more of that. If you want to create in red blood cells, again, more hemoglobin, or in muscle cells, more myoglobin, you create those proteins, right? And proteins, again, are made up of our amino acids. The other big organic compounds we sometimes pay attention to are nucleic acids, which then come together to create DNA and RNA. For the purpose of this slide, we're not quite going into too much on the nucleic acids and that regulation, since that kind of plays into more about cell cycle, cell signaling, and reproduction. Okay, so kind of to put all this again, what are our main organic compounds that our digestive system works really hard to take in when we ingest and chemically and mechanically break down foods? We work really hard to bring in individual fatty acids, monosaccharides, and individual amino acids. And depending again on the cell's state, the hormones, the fed versus fasted indications we have, when cells intake those mono or simple, very micro kind of molecules, depending on the state, depending on the hormones, depending on the status, that could lead to anabolic kind of building bigger molecules, or catabolic breaking them down using the energy stored between the carbon-carbon bonds to create more energy. All right. All right, so again, we primarily really like to use glucose, our carbohydrates, to help us generate our ATP in many of our cells. And the reason why is because glucose, in breaking it down completely to 6 carbon dioxide molecules, we create ATP a little bit up front with glycolysis and then a lot on the back end. All right, so the breakdown of glucose completely to 6 carbon dioxide molecules. So the 6 carbons in glucose become 6 carbon dioxide molecules. We first do a split in half of glucose to pyruvate through the cytosol, through the cell, cytoplasm, outside any organelles, with the use of a bunch of protein enzymes, and we create, again, a split in half, so 3 carbon molecules pyruvate. We generate a little bit of ATP. This is the only time we generate ATP without oxygen being present, and it's one of those reasons why, especially in muscles, we can create or maintain some high energy, high activity and intensity output when we've kind of reached our max on what we can create ATP-wise with our oxygen and our mitochondria, okay? And the high amounts of pyruvate can accumulate, and if we run out of those NAD molecules, we start to take pyruvate and convert it to lactate. So again, this process, keeping it going, can sometimes be why in exercise we achieve lactic acid thresholds, okay? Ultimately, the 3 carbon molecules of pyruvate need to go into the mitochondria. In doing that, they create a 2 carbon molecule called acetyl-CoA. Acetyl-CoA gives us a little bit of energy and liberates one of those carbon molecules as carbon dioxide. That first step in, then our acetyl-CoA joins a 4 carbon, so 2 plus 4 equals 6. It's known as the citric acid cycle because the 2 plus 4 binding together forms citrate. That's the first molecule in the cycle. Another name for it is just the Krebs cycle. All right. Mostly what we're going to do in the Krebs cycle is we're going to create a little bit of ATP. We're going to create much more NADH, FADH2s, which represent protons and electrons being pulled and held temporarily until we can then transport them in a manner that helps us through oxidative phosphorylation and binding 2 protons and 2 electrons with water molecules together or with oxygen molecules to create water. And in doing so, we then take the rest of the energy. Some of it's lost as heat, and some of it then makes more phosphate-phosphate bonds. Okay, so that's complete breakdown of a simple sugar, glucose. All right, and glucose is one of our preferred molecules to use for energy making, especially in the nervous system. All right, most of your other organ systems can use glucose, but can be told to not use glucose or told to use other sources primarily depending upon hormone and status of glucose levels in our blood. Okay, to take a little more of a in-depth look at what is glycolysis, you do not need to know the steps, but know that in this picture, let me see if I can get, all right, you start with 6 carbons. 6 carbons become 2 molecules of 3. So you would think like, oh, I just cut it in half. I take the energy and I make some ATP. Of course, it's not that easy. So this process is 7 steps as shown here. They take place all within the cytoplasm of the cell. There is some ATP expended. There is some ATP created. But if you take, again, like in accounting terms, if I put 2 molecules of ATP in and I get 4 total out, I only net a total of 2. All right, I net NADH molecules, which later are going to help me make more ATP. But up front, without oxygen being available, I net 2 ATP. I do all of this considered anaerobic metabolism because it's without oxygen. It's all taking place in the cytoplasm of a cell. Okay? Now, eventually, once I get pyruvate, I need it to go into the mitochondria. This little reaction right here, where I'm going to take coenzyme A, attach it to pyruvate, but I'm liberating a carbon dioxide, making more NADH, forming a 2-carbon molecule known as acetyl-CoA. That's part of how I get pyruvate into the mitochondria. If pyruvate really accumulates, remember, to keep more NAD available, I'll start taking pyruvate, adding hydrogens, and I'll make lactic acid. Right? And that can lead to, again, that buildup of lactic acid when we're exercising. That kind of puts us towards that lactic acid threshold. 2 carbons of acetyl plus a 4-carbon molecule makes citrate. So this is sometimes called the citric acid cycle. It's also sometimes known as the Krebs cycle. The end result is this cycle gives me a little bit of ATP, but it mostly breaks the rest of these carbon-carbon bonds, and it takes energy in the form of 2 protons and 2 electrons and puts them with NADH and FADH. I get a lot of those molecules where eventually I put them in my oxidative phosphorylation electron transport system. Right? And for that system, again, I'm still in the mitochondria. I'm on the inner membrane. I have some specific proteins embedded between the outer kind of area and the inner area. And I'm going to hoard and put a bunch of hydrogens up here. I'm going to carry the electrons in the membrane, in the proteins. And then I eventually want 2 hydrogens, 2 electrons to come meet oxygen, and in doing so, I create water. When I capture those energy molecules, I also put some of that energy in phosphates becoming part of ADP, so I create ATP. Again, because electrons are negative, I need pieces and parts of these protons or these proteins to have positive units, and positive units means I use iron and copper because iron and copper tend to be Fe plus or Cu plus. So they help, that's how I help carry the electron, which is a negative molecule. Again, some general things. You don't need to know the names of the specific steps or the specific parts of any of the cycles. You need to know the start and the finish. So ultimately, if I look at what is the breakdown of glucose, the breakdown of glucose in glycolysis is I take a glucose and I cut it in half. So I get 2 pyruvates, I get 2 ATP. All of this ends up in the cytoplasm of a cell. I get some NADH molecules. When I go further, pyruvate will eventually become acetyl-CoA and push into the mitochondria, join the Krebs or the citric acid cycle. A little bit more ATP, a lot more NADH and FADH2. All of those NAD and FADH2 molecules finish through the electron transport chain. I net 36 total ATP molecules, 34 being from what happens in the mitochondria. 36 comes from, again, that 2 that I got earlier with my breakdown of glucose to pyruvate. Okay? Now, sometimes I don't have glucose available, whether it's you're following a low glucose, an Atkins diet, a low carb diet, but my nervous system has to have glucose. So there is going to be some materials, some molecules that I can utilize to help me make glucose, especially when my diet is not bringing glucose into the system. All right, so typically when glucose is not available, glucagon is going to be released from the pancreas. That hormone is going to push the liver, so this happens in the liver, to make glucose. So gluconeogenesis. So genesis tends to be when we're making a molecule, we're making a new molecule of glucose. So that tells you in the name what we're doing. All right. Now, when we broke glucose down to pyruvate, okay, one of the 3 carbon molecules is sometimes pulled out to make glycerol, and glycerol serves as our backbone for fats. So anytime we start breaking triglycerides down, if we take some of the glycerol backbone molecules, we could potentially take the glycerol, put it back through the process of glycolysis backwards and create glucose. So if you are following one of those Atkins or super low carbohydrate diets, chances are some of the glucose you're making in the liver is from the glycerol when you're liberating triglycerides through beta oxidation. All right. We can also make glucose from pyruvate and lactate. So when I'm exercising heavily and I have a bunch of lactate accumulating and I start kicking the lactate into my blood supply and it starts making my blood pH drop and I become a little metabolic acidotic, some of that lactate when I'm stopping or done exercising, when I've hit my anaerobic threshold, can be reconverted back to pyruvate and ultimately back to glucose. So I can recover some of my glucose that I broke down in exercise from lactate. Another way to get glucose, not from a fat, not from, again, just recovering it back from the pyruvate lactate I previously broke down from previous glucose molecules, is I can create it from amino acid. And the amino acid I like to use is alanine, because alanine has 3 carbons. So alanine, if I take the nitrogen NH2, the amine group off, I can then convert it quickly into a 3-carbon pyruvate and then pull it back towards glucose. All right. Are there some other amino acids I might be able to pull in? Yes. Are there some other ways and molecules I might be able to pull into some of these intermediate steps? Yes. But my preferred way to make glucose, if I am following some type of situation of low glucose in my bloodstream, low glucose available, is I'm going to try to take glucose from some of my glycerol molecules when I break down triglycerides. I'm going to try to recover some of my lactate pyruvate back to glucose, and I'm going to convert some of my 3-carbon amino acids by pulling off the amine, the nitrogenous group, and converting it back to something like glucose. If I take pyruvate and I stick it in the, again, mitochondria, and I make it acetyl-CoA, so I go from a 3-carbon to a 2-carbon, and I get rid of carbon dioxide. Once I go to that 2-carbon molecule, I cannot go back. And this is part of the reason why people who eat lots of carbs, so let's say you're going to try to be vegetarian, and so all you eat is bread. You can still gain body weight as fat mass because all of that excess glucose and all of that excess pyruvate can then become acetyl-CoA. Acetyl-CoA cannot be converted back to pyruvate. So once I start accumulating all this acetyl-CoA, my only option to store it is to convert it into fat. Okay, so that's why people who eat too much glucose, too many sugars, too much carbohydrates can still, and very little fat, can still become fat and obese, because some of that glucose converted to pyruvate, converted to acetyl-CoA, will have to be as a 2-carbon excessive molecule stored as fat because it can't come back into pyruvate. Okay, now again, depending upon my hormones, depending upon the cell, depending upon the situation, if I want to take glucose and convert it into the stored form of glycogen, I would be making glucose to glycogen. So that's glycogenesis. If I'm exercising or if I'm starving myself and trying to burn some of my stored energy as glycogen that's broken back into glucose is known as the process of glycogenolysis. So anytime, again, genesis, we tend to say we're building. Anytime we're lysing, we're probably breaking. Okay, so glucose in certain cells, so this can happen in our liver, this can happen in our muscles, can be converted to glycogen and stored. Now, when we store glucose, which it's basically like a glucose molecule, and then you put an oxygen, and then you store another glucose molecule, and again, you store you have an oxygen connecting them. All right. So when you do glucose, glucose, glucose together, you start to create this long chain of glucose molecules. That's glycogen. This is still considered a carbohydrate. So as I store glycogen, I'm going to have to retain more water because this solute through osmosis is going to make me retain water. So when you watch a show like The Biggest Loser and they're calorie restricting and they're burning through their stored glycogen in their muscle and their stored glycogen in their liver to liberate glucose molecules so they can use glucose for energy molecules in their cells and to keep their blood glucose levels normal so their brain can use glucose for energy. What you're going to do is you're going to burn through your stored glycogen. And when you take away all that glycogen, you end up dumping a lot of water. And so that's why when people tend to go on a diet and they lose like 10 pounds a week and you say, oh, that's just water weight, where's that water? Well, here's that water. This is the water that you retain to help you in your muscle cells and in your liver cells store glycogen. So when you get rid of the glycogen, you get rid of the water. The first week of usually a diet is your body burning through its glycogen and then dumping its water that it held the glycogen suspended in the cells as a stored solute. Okay? So that kind of completes that. All right, so the end result is for carbohydrates. If I start with glucose as my simple molecule, if I completely break it down to six carbons to 6 carbon dioxide molecules, I will get myself in that process of glycolysis, citric acid cycle, and electron transport oxidative phosphorylation, 36 ATP. All right. The part of breaking glucose down in the cell's cytoplasm without oxygen is known as glycolysis. It doesn't require oxygen. It's still about 7 steps. All right, it gives me 2 ATP. Everything else for those molecules of pyruvate is going to take place in the mitochondria. First step is the Krebs or citric acid cycle, then eventually the oxidative phosphorylation, the electron transport chain. Ultimately, I'm going to need oxygen because in making the ATP molecules, capturing energy in phosphate-phosphate bonds, I need the energy of NADH and the hydrogen and electron of those molecules to be absorbed by oxygen and create water molecules. Okay. If I have a lot of glucose and I'm in a very big fed state, I'll probably have insulin around. Insulin is going to help push my body to store some of my glucose as glycogen. That's known as glycogenesis. When I don't have a lot of glucose available, and again, my blood glucose levels need to stay pretty constant, so my nervous tissue always gets energy, I can, again, with glucagon's assistance, push some of my glycogen to be broken down to glucose. That would be glycogenolysis. Some of that glucose can be used for energy in some cells. Some of that glucose will be liberated to the blood supply, keeping my blood glucose levels near normal or steady state, and then picked up by my nervous system. Okay, if I burn through my glycogen and I now need to continue to put glucose in my blood supply and I no longer have stored glucose available, I'm going to have to start making glucose. That's known as gluconeogenesis. The best way to make glucose is to revert some of my lactic acid back to pyruvate and eventually back to glucose. If that's not an option, I can also take some of my glycerol, which are 3 carbon molecules that I made from breaking down glucose previously, and then I attached them to fatty acids and maybe a phospholipid. Well, I can take that glycerol when I take the fatty acids and the phosphates off, and I can re-put it back through the back door of glycolysis and bring it back into becoming a glucose molecule. Right? It'll take 2 glycerols to do that because I need 2 3-carbon molecules to make 6. Alanine is also an amino acid I could take and push in along with another 3-carbon molecule and eventually recreate glucose from. Okay, so those are some of my best molecules to recreate glucose when especially I don't have any more glucose stored as glycogen in my liver and my skeletal muscle cells, and I'm not taking any glucose from my diet, so I'm in a very extreme fed or fasted state, and so I need to make glucose to keep producing or keep my blood sugar levels normal so my brain continues to stay normal. Okay, so that is carbohydrates. All right, let's stop here and stop the recording, and then I'll pick up with a second recording for lipids, but that way it'll kind of break this into smaller units. All right.